Welcome, and thank you for attending the Precision Nanosystems Tea Time webinar series. So today we have Professor Sophia Antimisiaris, who's joining us from the University of Patras, and she'll be discussing new approaches for targeted delivery, drug delivery with nanomedicines. Just as a refresher for who your Precision Nanosystems team members are, we have myself, AJ, and Richard that are based out of the UK. And then we also have Jürgen and Martin that are based out of Germany. So if you do have any other questions, either from this webinar or any of our other technologies, please feel free to reach out to us. So what I'll do now is I'll hand it over to Sophia. Thank you, Sophia.
Hello, I want to thank you for the invitation. I will now go to the presentation. As I already said, I'm going to present some work about new approaches for targeted drug delivery with nanomedicines. I'm going to go very fast through the introduction and the type of nanomedicines, and then talk about their advantages for drug delivery, what we have accomplished up to today, and what are the challenges and potential ways to overcome the challenges. I'm going to go pretty fast in the introduction part, so if there are any things you want to answer, please come back to me about this. If we want to give an introduction, we can say that nanomedicine is a branch of medicines that seeks to apply nanotechnology for prevention or treatment of diseases, for imaging, diagnosis, monitoring, and for repairing or regeneration of biological systems. Actually, nanomedicines are either drug carriers, imaging devices, vaccines, or devices for tissue engineering. We will focus today on drug carriers. And the nanomedicine market is pretty big, and it's expanding with the years. The projection for 2027 reaches close to 400 billion. So we can understand that it's a big market. Now, the types of nanomedicines for drug delivery can be of various origin. We have organic and inorganic, and of various geometries and shapes. What is important in this case is that they obey to the requirements that we need to have in order to use them as drug carriers. First of all, they have to have very high drug loading. They have to be fully biocompatible if possible. They have to have nanosize, which they would retain during their journey to the site of action. They have to be able that we can surface, tailor their surface, and they have to have an easy manufacturing under GMP compliant conditions. And this is where the systems from Precision Nanomedicines come in. We are recently using one of their equipment, and we are very pleased with it. But unfortunately, I don't have results to show today because we have not published any yet. What I want to say now is that we will focus on liposomes, because this is what we work with in our laboratory. They have several advantages, but the main advantage is that they are in use in the clinic for almost 30 years now. So this means that they are a tested system. They have some disadvantages which we can solve with by preparing hybrid systems. Now, how can they improve or advance the therapeutic performance of drugs? First of all, by protecting the drug from harsh environments, either at the site of administration or at the site of action, and also protect the site of action, the cells over there, from very high concentrations of drug, which in some cases may be toxic, by controlling the drug release from the system. They can also modify the pharmacokinetics and target specific sites, and this way they increase therapeutic effect, decrease side effects, increase bioavailability. And when we have all this, of course, we will have a reduction in the cost because we just have to give less amount of expensive drugs. Now I will show some examples from our work through the previous years, how liposomes can protect, or these kinds of nanomedicines can protect molecules, as for example, the protection of curcumin from chemical degradation, how we can control or sustain the release of drugs. And further down, I will give examples about targeting. Here we see some example about the protection of curcumin degradation in serum at 37 degrees. And we see that if curcumin is plain, or even if it's incorporated in cyclodextrin complexes, it degrades very fast, as we see in this left side graph. But when we go here, we see that liposomes can expand the period that curcumin is stable in serum, and we can have a very high increase in bioavailability. So this is one example. And now we go to some examples about sustained release. We can sustain the release of drugs by incorporating them into liposomes and incorporating the liposomes then in different types of hydrogels, which may be thermosensitive or pH sensitive, and they can also be in situ forming. So here we see the example of dispersing the drug itself in, which is calcium in this case, in gels, whereas when we put the calcium in liposome and put the liposomes in gels, as we can see, we can have a slower release from the system. This is from some earlier work that we did in our lab. Another example is with this pH sensitive gel. And when we have the plain liposomes, we see that calcium is released quite fast. At 5 days, we have almost a zero order, but 100% release. But when we put the liposomes in the polymer, which is a pH sensitive actually polymer, we can control the release, which is zero order, but we can control by manipulating or using different concentration of polymer. So we also have some examples from our lab when we use liposomes as drug delivery systems on surfaces. So we can immobilize them on surfaces, in this case, metallic surfaces, and increase the hemocompatibility in case we want to put them on metallic stents, let's say. So here we see that the hemocompatibility of these surfaces was increased highly when we added heparin in the liposomes and immobilized the liposomes on plasma-treated surfaces. Another example, similar, is when by adding topramycin liposomes on surfaces and making these surfaces have antimicrobial properties. So we see that the bacteria cannot adhere at these surfaces, whereas in the control surfaces, we have high adhesion of bacteria. Now let's go into the actual accomplishments in the field. We have many liposomal drugs and other nanomedicine drugs in the market today, but most of them work with the EPR effect, which maybe you know, it's the effect that small particles, nanoparticles, can go at higher extent to cancer cells because of the leaking vasculatures that they have in tumors. So this way, we may say that we have actually passive targeting. When we want to have active targeting, we have to have ligands that are that have receptors that are overexpressed on the target cells in order to have a higher and active targeting system for our cells or tissues. But when we ask the question, has active targeting been actually translated in the clinic? The answer is no. We see that we have a lot of FDA-approved nanomedicines. However, the ones that are actively targeted are still in clinical trial phase, and they are not translated. This can be attributed to the proteins that adhere on their surface when they circulate in the blood, the protein corona, as they say it, and several other factors have been implicated, like the expression of receptors, the location of the proteins on the cells, the size, the shape, the charge of the nanomedicines, the ligand density, the internalization medicine, many, many factors. However, up to today, we could not overcome these problems, and we have a very huge bottleneck in the field. So I'm going to show you some examples about ways to overcome this bottleneck. The first couple of ways are by increasing the targeting potential. And I will also show some other approaches as exploring different routes of administration or getting inspired by nature. Now, to increase the targeting potential, we can add on the nanomedicines different ligands that are targeting different receptors. So at the same time, we can target 2 or more receptors with one nanomedicine nanoparticle. Or we can add an additional function as magnetic field or ultrasound. There's a lot of research in the literature today, but I will show you what we have done in our lab. So to target the blood-brain barrier, we have used these liposomes that have a transferrin antibody to target the transferrin receptor, and another peptide ligand to target the apolipoprotein. So we have them both on the same nanoparticle liposome, and we see in vitro, in this in vitro model, that we have, of course, a higher transport when compared to our control when we have one of the ligands on the liposomes. But when we have both of them, this transport is increased dramatically. And this we also saw in the in vivo experiment. So we see that this is an approach that we can use. Another approach is to use an extra function and prepare what we call multifunctional medicines or liposomes in this case. By adding, by encapsulating magnetic nanoparticles in the center of the liposomes, we have succeeded to have attraction by magnetic field. And we see in this graph that when we just have the targeted liposomes in these cancer cells, we have a reduction compared to the non-targeted liposomes in their viability. But when we also use a magnetic field and we have the magnetically labeled liposomes, we have a further reduction. So this is a way to improve, again, the targeting potential by guiding the nanomedicines to specific sites. But sometimes it's not so easy because the cells we want to target are located deep in the body. So it's not something that can be applied in any case. Now, going to the other approaches, one of them is to explore different routes of administration. The other is to get inspired by nature. So I will start with the first, and I will start actually with ocular delivery. Nanomedicines have very good applications for intravitreal injection of the drugs that have to stay for longer periods because the injection is not so easy to do. So you want to decrease the frequency of injection, and you want your drug to stay there bioavailable for a long period of time. So when we prepared these, in the one system that we actually developed was to put the liposomes in a hydrogel. And in this case, it was a thermosensitive hydrogel, Puronic F127. So when we have the drug by itself, we see that in vivo, it goes away very fast. It clears from the vitreous after intravitreous injection. When we have it in hydrogel, it's a little bit better because it stays there a little bit more. Liposomes behave a little bit better than hydrogel, but not significantly better. But when we have the liposomes in the hydrogel, the drug in liposome and in hydrogel, we see that we increase very much the bioavailability of the drug, and also we had a better result in the action of the drug. And another system that we developed is this Liposome Aggregate platform, which, as we see over here, is actually composed of negatively charged liposomes, which are mixed with a positively charged molecule. In this case, we used protamine, and we get this kind of aggregates. And when we do this and inject this kind of systems for a non-steroidal anti-inflammatory drug that is clearing very fast from the ocular cavity, we see that we can get a huge increase in the bioavailability, and this is seen for longer time points over here. What was very important was also that by confining the drug, the high concentration of drug in this lab system, we had a decreased inflammatory effect in the ocular tissues. So this was also a very interesting and important effect, advantage, let's say, of these kind of systems. So we see that if we go to topical application, we may have better therapeutic effects. Another route of administration that we tested in the past is the intrapleural injection. In this case, we saw that when we inject intrapleurally liposomes, they stay in the cavity for long periods of time. Up to 24 days, we have very high signal. But when we go, and this can be controlled by different lipid composition of the liposomes. But the most important was that when we injected with these type of liposomes, non-tumor inoculated and tumor inoculated mice, while in the first case we saw that some hours after injection, we have a lot of drug going out and going to the liver and leaving the lungs. When we worked with the tumor inoculated ones, we see that a lot of the signal is actually in the tumors, and some is still in the lungs, maybe in tumors that we cannot really separate from the lung tissue because they were very difficult to separate. This was very interesting, and it shows that we may have a very high advantage when we use this route of administration for diseases in the lungs, such as MPE, which stands for malignant pleural effusion. And this actually explains why, when we injected this drug, which is a KRAS inhibitor named delta-racin, with a good therapeutic effect, as we saw in this publication for MPE, we could have the same effect after one only dose of intrapleurally injected liposomes compared to 14 daily doses of the free drug. So this was actually a very surprising and good effect for us. And we see the potential of these kind of topical injections of nanomedicine products. Now, the other part I wanted to talk about was about getting inspired from nature. You may have heard about extracellular vesicles and exosomes and how to use them in drug delivery. Many people are trying to do that now. It's a very hot subject in the drug delivery field. I have to say that exosomes are actually vesicles that are prepared, that are developed in the cells, and they go from one cell to another, transferring the compounds between them. And in some cases, the two different cells are located in far apart tissues in the body. They are also implicated in metastases from one tissue to another of cancer. So somehow they manage to get into the cancer cells or into other types of cells. So it's very important to see if we can use them for drug delivery. The problem is that there's a very low yield when you try to isolate them. So some people propose to use whole cells to prepare vesicles because the surface of the exosome is actually similar to the surface of the cell. When we see what the exosomes are composed by compared to liposomes, we see that they are very similar, only that the liposomes usually have a PEG coat to avoid uptake by the RES and fast accumulation in liver and spleen, while the exosomes have a lot of proteins in their bilayer, and this is what guides them probably to the specific cell types that they have organotropism for. So what we try to do here is to see if we can use these kind of cellular vesicles to target the brain. And we prepared vesicles from different types of cells, from the same brain cells that we used in this in vitro model of the blood-brain barrier, from cancer cells, B16 cells, and also from normal cells. And when we tested their interaction with the blood-brain barrier cells, we see that their uptake is much higher when they are derived from the same cells compared to the cancer cells. And the cancer cells are not very bad, but they are much better than the normal cells. And when we compare these with liposomes, we see that we have a high increase and we compare them with targeted liposomes. In some cases, we see that there is a significant difference, especially in this case. And also, we saw the same results when we did this monolayer study, transport study, and this effect was also confirmed by FAC study. So when we went in vivo, unfortunately, we saw that these cellular vesicles are rapidly taken up by liver and spleen. This is something known in the literature, so there is need for engineering. So we pegylated them by specific methods that we use also to pegylate liposomes. And when we did this, we saw an increase in the amount that we have in the brain. So the brain signal increased when we pegylated. And what is very important is that the brain over liver and spleen signal that we got in the ex vivo results after extracting the organs from the mice were very high. In the case of these cellular vesicles that were composed from the brain cells, compared to the B16s, which were not very low, they were also high. And it's important to compare these values with what we get in the case of targeted liposomes. For the dually targeted liposomes, we get from 0.3 to 1.6 ratios, or for 0.6 to 1.5. And in this case of the CPV, if they are not pegylated, we get 4.9. If they are pegylated, we get higher than 10. So we can see that there is a very high potential for these systems, these kind of systems, to be used as nanomedicines for drug delivery applications. So I will wrap up now by summarizing. I hope that I convinced you that nanomedicines can protect drugs from degradation, can prolong their retention at injection sites, can increase the topical drug bioavailability, and also to increase your knowledge that today there are still several challenges. The main challenge is active targeting and bypassing biological barriers with actively targeted systems. And the potential solutions that we are investigating in our laboratory are the ones that I showed you before. And we are particularly interested in this time in the exploration of alternative routes of administration of nanomedicines, which have not been fully explored up to date, and about systems inspired from the nature and from our own bodies, as from the organotropism of extracellular vesicles. So this is why we are further exploring the scenario of using such cellular vesicles for drug delivery applications. Ending, I would like to thank all the contributors that work of which I showed today, which are shown over here from my lab. Also some collaborators from the Department of Medicine of our university and from the University of Crete, where we work with a group of Professor Tilibaris for the ocular delivery. And of course, some people from other collaborating programs that we worked with them for providing some specific compounds. And of course, the funding. Thank you very much. And I am open now to hear any questions and try to answer any questions that you have.
Well, thank you very much for that very interesting, very enlightening talk on a, well, very important and critical field. So, and thanks everyone also for attending. There will be another Tea Time webinar in 2 weeks' time at the same time. That one will be presented by Samir Cherakui of Bracco from Switzerland. So thanks to everyone once again for attending, and we look forward to seeing you next time. Thank you.
Thank you.